In this topic, we're going to have a look at gene technology, an introduction, and then we're going to look at how to identify and isolate genes. So by the end of this topic, you should be able to answer the questions: what is gene technology? What is insulin? What are the steps to make insulin? And then we're going to look at how to identify and isolate genes using reverse transcriptase and restriction endonucleases. So what is gene technology? Well, this allows for genes to be manipulated, altered, and transferred from organism to organism. So it's also called genetic engineering. And it's allowed for the characteristics of an organism to be changed, either by directly altering its genes or by transferring into it genes from another organism. So this means that the affected cells will make a different product during protein synthesis. So if you have a look at this bacterial cell here, you can see the DNA is a loop represented by that blue round circular loop. If you insert another gene into this bacterial DNA, when this bacterial DNA undergoes transcription and translation, your gene is also going to undergo transcription and translation. So you'll be able to get your desired protein. Now you've heard about the hormone insulin, which is a protein, and people who have diabetes mellitus cannot synthesize insulin in their pancreas, so they need to get regular injections of insulin. Now until recently, this insulin was obtained from the pancreas of pigs or cattle. This extraction was expensive, and many people didn't like the idea of using insulin from an animal. The insulin from cows and pigs is also not identical to humans, so it can have side effects. In the 1970s, biotechnology companies decided to work on the idea of inserting the gene for human insulin into a bacterium and then using this bacterium to make insulin. So it was in the 1980s that they succeeded, and this was all due to gene technology. So here's a simplified diagram of how it happens. You take insulin from a healthy person, you extract the gene, you insert that gene into bacterial DNA, and then your insulin protein gets made when that DNA undergoes transcription and translation. So what is insulin? Well, it's a small protein, about 51 amino acids, and it's got an A chain and a B chain. So the A chain comprises 21 amino acids, and the B chain comprises 30 amino acids. And these are held together by disulfide bridges, as you can see represented by those yellow S circles. The gene for insulin is found on chromosome 11, and it's only got 153 nitrogenous base pairs. This means it's got 51 codons. So how many amino acids are there? There are 51 amino acids. Right, let's have a look at the steps of making insulin using DNA technology. The first is to identify and isolate the DNA fragment that has the gene for the desired protein. Then you clone the DNA using DNA polymerase. You insert the DNA fragment into a vector. And then transformation is the transfer of the DNA into suitable host cells. We then need to identify the host cell that have successfully taken up the genes, and we use gene markers to do this. And finally, you grow up the population of host cells to produce your protein. Right, let's have a look at the first step, identification and isolation of DNA fragments. So before a gene can be transplanted, it must be identified and isolated from the rest of the DNA. Given that the required gene may consist of a sequence of a few hundred bases amongst the million in human DNA, this is quite an intricate process. So there's two methods that you can use. Either reverse transcriptase or restriction endonucleases. And both of these involve enzymes. Okay, let's have a look at reverse transcriptase. Retroviruses are a group of viruses. The one that you've come across is HIV. So retroviruses have their genetic information in the form of RNA. And here you can see a virus infecting a cell. So that virus is that little green circle. Notice how the virus has got RNA. Its RNA can then be made into DNA by an enzyme called reverse transcriptase. So you can see that process occurring. The viral RNA is pink, and then using reverse transcriptase, blue DNA is being made. This DNA, that blue DNA, is then incorporated into the DNA of the host cell. So more viral RNA is made when the cell's DNA undergoes transcription. So scientists can use this process of reverse transcriptase to isolate a gene, for example, the gene for human insulin. So what happens is the beta cell of the islet of Langerhans in the pancreas is selected, and the mRNA that codes for insulin is extracted. Reverse transcriptase is then used to make DNA from RNA. This DNA is known as cDNA because its nucleotides are complementary to the mRNA. So you can see that cDNA is the purple strand. Then you take that cDNA and you use it to make a complementary, or use it as the template to make another strand of DNA. And you use DNA polymerase to do this. So DNA polymerase is the enzyme, and it builds up the complementary nucleotides of the cDNA template. Now you've got a double strand of DNA, and this is your required gene. So here's a nice diagram to illustrate what I've just discussed. You've got your mRNA that's been isolated from the cells. You use reverse transcriptase to make cDNA. And then you take that cDNA and you make another strand of DNA so that you've got your desired gene. Okay, let's have a look at restriction endonucleases. Now, bacteria are frequently invaded by viruses that inject their DNA into them in order to take over the cell. So some bacteria defend themselves by producing enzymes that cut up the viral DNA. And these enzymes are called restriction endonucleases. And many types of restriction endonucleases exist. Each one cuts a DNA double strand at a specific sequence of bases called a recognition sequence or a restriction site. So sometimes this cut occurs between opposite base pairs, and this leaves two straight edges known as blunt ends. For example, one restriction endonuclease cuts in the middle of the base recognition sequence GTTAAC. Other examples of restriction endonucleases that produce blunt ends are ALU1 and HAE3. Now, other restriction endonucleases cut DNA in a staggered fashion, and this leaves an uneven cut in which each strand of DNA is exposed, and it's got unpaired bases where it's exposed. So an example is a restriction endonuclease that recognizes a 6-base pair sequence AAGCTT. So in this figure, look at how the unpaired bases are the same on the bottom and the top. So if you read the 4 unpaired bases at each end from left to right, they're opposite to each other. So this is what we call a palindrome. So we say that they are on palindrome. The recognition sequence is referred to as a 6-base pair palindromic sequence. And this feature is typical of the way restriction endonucleases cut DNA to leave sticky ends. Other examples of restriction endonucleases that produce sticky ends are BAMH1 and EcoR1. So here you can see how the different enzymes recognize different base sequences. Notice how you get blunt ends at the top and sticky ends at the bottom. So some enzymes you might come across are EcoR1, this produces sticky ends, BAMH1, now sticky ends allow another piece of complementary DNA to be joined. So if you have molecule A and molecule B, you digest them with restriction endonucleases, or the same restriction endonuclease is used. This will prepare the gene of one and it can be inserted into another piece of DNA because their sticky ends are complementary. And when you join this gene with the other DNA, we call this recombinant DNA. So in summary, what have we looked at? Well, we discussed gene technology as manipulating or inserting genes into organisms. And remember that insulin is a protein that's got 51 amino acids. We went through the different steps to make insulin, so identifying the gene to be isolated, cloning the DNA, and inserting it into the vector, and then transformation and identification. And we looked at 2 methods of identifying and isolating genes using reverse transcriptase. So remember that you make cDNA from mRNA in that case. And then restriction endonucleases. So things to remember about restriction endonucleases that you get blunt ends and sticky ends. And that concludes our lesson, the end.